for my opinion, very nice quote by George Box, which sounds like all models are wrong, but some are useful. And I think it's in particular very relevant to the computational studies that we are all discussing during this training school. And here on the slide, you can see the short outline of my talk. So I would like to maybe shortly, I would say, discuss the structure and dynamics of ion hydration shell, which is forms around the DNA. A bit talk on the ion DNA interactions and slowly move to the results that we recently obtained on the vibrational dynamics of the DNA ion water systems, and also to talk about the ion-mediated DNA association. So let me start from there. Could you please switch the slide? Thank you. Let me start from the beginning, from the one of the main object of this talk, from the DNA. And DNA is the central molecule for all the living organisms. It's a long polymer which consists of the repeating monomer units, which called nucleotides. And nucleotide, it consists of nitrogenous base, which is linked to the sugar ring. It's not really visible that is the ring, but believe me, it's a sugar ring, and also to the phosphate group. So there are four types of the bases. It's adenine, thymine, guanine, and cytosine. So DNA usually consists of two polynucleotide chains that twisting around each other to form the double helix. And the sugar, together with phosphate groups, they form the backbone of the DNA, which is placed outside of the double helix. And inside the bases, they form complementary pairs through the hydrogen bonds. And the adenine always pairs to thymine, guanine always pairs to cytosine. The surface of DNA, as you can see, it's not uniform, and the two distinct regions can be distinguished. It's a major and minor groove. However, DNA, it doesn't exist in the isolation, and the important part of its structure is the surrounding environment, and in particular, its water molecules and ions, which is key factors to maintain the stability of the double helix. And importance of hydration, it became evident starting from the very first X-ray diffractional studies of the Rosalinda Franklin and Raymond Gosling. So in their experiments, they exposed DNA to two different levels of humidity. And in a lower humidity, DNA took more compact conformation, producing one diffractional pattern shown here. And when the humidity increased, DNA became more extended and produced the famous X-shaped diffractional pattern. So these experiments, they showed that DNA can exist in different conformation. In particular, here it's A and B, but there are many other conformations, but we will not discuss them now. So DNA double helix can form where the sufficient number of water molecules are present around. And here on the slide, I wanted to summarize the known data on the level of humidity and on the number of water molecules per base pair, which are needed to DNA to take some of the conformation, but take in mind here is the results mainly for the B form of the DNA. But what is important here is that the number of water molecules and obviously the level of humidity, it strongly depends on the type of the counter-ions which is present in the sample. And so in general, the larger ions, they require less water molecules to stabilize the double helix. But there is the one exception. It's a lithium ion, which is the smallest from this list, but it can somehow effectively neutralize the negative charges of the DNA. And as a result, the less water molecules are needed. So the water molecules together with ions, they form around the DNA ion hydration shell. that extended more than 80 angstrom from its surface and can be roughly divided into several layers, where the outermost layer, it behaves more like the bulk solution, closer to DNA but without the direct contacts with the surface. The intermediate layer forms where also the ion atmosphere is absorbed, but I will be back to it later. And water molecules together with ions, which is in direct contact with the DNA surface, they form the innermost solution layer, which is the most ordered part of the hydration shell. And interesting feature of the B-form DNA is that the water molecules in its minor groove, they can form the ordered structure, which known as a spinal hydration, where water molecules, they can bridge the atoms of the bases of opposite chains of DNA. And these water molecules, in turn, they can form the extended hydrogen bonding network with the neighboring water molecules, and as a result, the characteristic hexagonal shape structures of water molecules can appear in the minor groove. And actually, for the very long time, it was believed that these structured forms of the water are mainly limited by the narrow minor groove. But recent study, which combined the chiral sun-frequency generation spectroscopy together with MD studies, they show that actually DNA can structure water molecules in entire first hydration shell, including the major groove, minor groove, and also the area around the phosphate groups. So here on the graph, you can see the water molecules from the first hydration shell that's shown in red, and the from second hydration shell that's shown in blue. And the calculated spectra here, it shows that the water molecules from the first hydration shell, they produce the dominant contribution to resulting overall signal. And this indicates that the around DNA, the highly structured forms the water molecules form the highly structured arrangement. So yeah, in addition to the structure of hydration shell, its dynamics also not uniform and strongly depends on the position of water molecules around the double helix. And this figure shows that the hydration is highly dynamic, and water molecules, they continuously change their position within one hydration layer and also exchange between the different hydration layers. And both experimental and the computational studies, they show that characteristic residence time in the case of the bulk water equal around 1 picosecond, while closer to DNA, the dynamics of water molecules, it's notably slowed down. And near the phosphate groups, the residence time of water can reach 10 picoseconds, while the slowest water molecules, they're absorbed in the inner region of the minor groove. It's a major groove, where the residence time can reach 100 picoseconds, sorry. And the slowest water molecules absorbed in the case of the minor groove, where the residence time can exceed 200 picosecond and can reach even the nanoseconds. But yeah, let me go slightly back to the structure of the hydration shell, specifically to ion atmosphere that I mentioned before. And since the DNA is highly charged polyelectrolyte, it attracts the positively charged ions, which form the ion cloud around the double helix. And this was firstly predicted by theoretical studies, and later it was confirmed both the experimentally and NMD studies as well. So here I want to show the example of the MD study on the ion distribution around the DNA in the case of the sodium chloride solution. So the first graph shows the ion density as a function of the distance from the DNA axis. And you can clearly see that in the case of the sodium ions in this line, they are predominantly found near the phosphate groups of the DNA and can also be seen in the inner region of the double helix. While the chloride ions, they are depleted near the DNA surface and reach their bulk concentration in the larger distances. And the lower graph show the cumulative charge of ion around the DNA. And the dashed line here show the Manning radius. It's a distance where the approximately 75% of negative charges of DNA are compensated or neutralized by ions. And the calculations show that this distance is equal approximately to 16, 17 angstrom from the DNA axis, which is also in a good agreement with the theoretical predictions. So ion can interact with DNA in the different binding sites. And here shown the results on the concentration of the different ions around the DNA. It's a MD study. And this graph shows the top-down view on the DNA cross-section where the reference binding sites for ions are also shown here in the lower graph. So one can see that the distribution of ions around DNA is strongly ion specific, and for example in the case of the lithium ions the highest concentration is observed near the phosphate groups of the DNA, while in the case of the potassium or cesium ions they are predominantly found in the inner regions of the double helix. So these differences, they reflect the distant binding affinities of the ions, and therefore, despite they carrying the positive charge, alkali metal ions, they exhibit the different spatial distribution around the DNA. Another important question on how long these ions can remain near the binding sites of the DNA. And here there is another MD studies, this one, which show the residence time of the series of the different ions. It's potassium, or sodium, potassium, cesium, and magnesium, which is located in the different regions of the double helix. In short, the longest residence time were observed in the case of the minor groove of DNA, with the longest time for cesium ion, which can reach 1 nanosecond, and the shortest residence time were observed for the case of the sodium ion, which is less than 100 picosecond. In the major groove, the residence time are shorter if we compare it with the minor groove, with the longest time observed still for the cesium ions and some of the magnesium ions. And near the phosphate groups, the residence times of ions, they can reach up to 400 picosecond, and they follow this order where the magnesium ions have the longest residence time. So interactions of ions with the DNA is strongly ion specific, and particularly depends on the hydration properties of the ions. And depending on how the ions structurize water molecules around themselves, they often classified as structure making or the structure breaking ions. And structure-making ions, it's like the magnesium, lithium, sodium, etc. They order water molecules around themselves, and they have relatively stable hydration shells. And because of the dehydration is energetically unfavorable process, these ions, they often interact with DNA through the water molecules in their hydration shell. While in contrast, the structure-breaking ions, they have less ordered hydration shell, so they can easier lose their water molecules, and approach DNA closely, including the major and minor group. And I forgot to say that the key factor that governing these processes, these interactions, it's ion charge density, and ion with the higher charge density, then they generate the stronger electrostatic field around themselves, so they stronger attracts water molecules compared to ions with the lower charge density, like the lithium or rubidium, etc. So sorry, thank you, thank you. So the dynamics of the water molecules in the ion hydration shell, it also can be probed using the MD method. And the left graph show the residence time of water around the ions in the bulk solution. And here we can see the clear trend where the water molecules near the lithium ions, they have the longest residence time, tens of picoseconds, while the sodium ions, they also can retain water molecules relatively strongly. But for the potassium, rubidium, and cesium, we observed the lowest residence time for water molecules in the hydration shell. But the graph on the right, it also showed that the residence time of water molecules around ions, it strongly depends on the position of ions around the DNA. And in general, the residence time of water of ions near the DNA surface, it's longer than in the case of the ions in the bulk. And the longest residence time observed in the case of the water, which is located around the ions in the minor groove of the DNA. So one can see that both the ion time and its location around the DNA, it's influenced the dynamics of hydration shell. And among all the alkali metal ions, the lithium ions, they stand out by having the most stable hydration shell with the slowest dynamics. And thus we can see from the discussed above that all the elements in the DNA ion water system, they are closely interconnected. And to gain the deeper insights into interaction of this complex system, we also should study, or we can study, the vibrational dynamics, since the vibrational spectra, they consist all the contribution, all the signatures from the DNA, water, and ions, and can be very important to understand deeper the structure, dynamics, and interaction of this system. So to study the vibrational dynamics of the water ion DNA system, in the recent work together with my collaborator Hadi Azizi from ICTP, we performed the atomistic molecular dynamics simulation for the different system which contained one DNA macromolecule, which is immersed in the box with water. And we added there to different system, we added the different salts with different alkali metal ions, including the lithium, sodium, potassium, rubidium, and cesium. And so here I summarize some details on the force field that we used, but I will not stop on them now because they will repeat in the other simulation, so I will discuss them later. And to study the vibrational dynamics of modeled system, we first calculated the absorption spectra that can be obtained as the Fourier transform of the correlation function of the time derivative of total dipole moment. So please take in mind that the total dipole moment here, it presents as the summation of the different different dipole moments of the components of the system. It's a DNA, iron, and water, and those the absorption spectra, it's contain both the cross-correlation terms and after-correlation terms, but since in this work we were mostly interested in the individual contribution of each the system elements, we neglected the cross-correlation and studied only after correlations. So here's the results that we obtained for the DNA in the case of the system with the different alkali metal ions. The gray arrows below the graph, they show the characteristic spectral range where the characteristic vibration of atomic group of the DNA is observed according to known experimental data. So in general, our results show that there is no significant influence of the different ions on the vibrational dynamics of the DNA, but here is only one exception. It's a lithium ion where we see the changes in the spectra where the backbone vibration is observed. In particular, it's a stretching of the PO bonds of the phosphate groups. And to analyze this, we also studied the distribution of the ions around the phosphate groups of DNA. And our results show that lithium ions have the highest concentration near the phosphate group, which is consistent with all the results that I discussed before. We also calculated, thank you. So we also calculated the absorption spectra of all the salts that we studied, and we can see that the shape of the absorption spectra for the ions with the positive hydration, it's lithium and sodium. Their spectra are different from those for the ions with the negative hydration. And the positive one is characterized by 3 distinct bands for the lithium and sodium, while the other are characterized by only for 2 bands. And to validate these results, we compared them with the available experimental data. This is the Terker's spectroscopy, and I can say that we obtained the good agreement with the experiment. And also we can see that here the band near the 200 inverse centimeter is observed in the case of all the calculated spectra. And our analysis show that this band should associated with the vibration of fluoride ions, which is also in a good agreement with experiment. If you take a look to this spectra for lithium chloride, we can see the band near the 200 inverse centimeters, while in the spectra for lithium bromine, this band are vanished. But here we also faced the problem, and this is the problem that we cannot separate the contribution from the anions and cations from the absorption spectra, since the ions it's point charges. And therefore, we also calculated the vibrational density of states, which can be obtained is the Fourier transform of the velocity after correlation function. So we calculated those spectra for all studied ions in the case of the different region of the double helix. It's a phosphate groups, major group, minor group. And also we compared the spectra for ions near the DNA with the spectra that we calculate for the case of the system of bulk solution. So in general, our results show that the spectra for ions which is located near DNA surface, they are shifted towards the higher frequency range, which is most pronounced in the case of the minor groove of the DNA. And actually, we see their shift mostly for the case of the ions, for the structure breaking ions. It's rubidium, cesium, and sodium, and potassium, sorry. And what is also interesting from these results is that in the case of the spectra of lithium, which is located near the phosphate groups and the major group, we observe the additional band on the or the shoulder that is located near the 800 inverse centimeters, where also, according to experimental data, the backbone vibrations observed. And therefore, this bond can also can be assigned to the couplet vibration of lithium together with the phosphate group, which also provide the further evidence of the strong interaction of lithium ions with the DNA phosphates. So all the discussed results so far, they indicate that lithium ions, they have in particularly strong binding affinity to the phosphate groups of the DNA. And here I show the figure that I showed before on the lithium distribution around the DNA with the highest concentration near its phosphate groups. And additional insight into these interactions were provided in the earlier molecular dynamics study by Lyubartsev and Lansonen. And here is the figures from their study. It's the special distribution function of the water molecules near the phosphate group. So the water molecules shown here as a violet and blue, violet oxygen atoms, blue it's the hydrogen atoms, and the same special distribution function shown in the case for the lithium ions, shown in violet, near the same binding site. So one can see that lithium preferred position follows the hydration structure of the phosphate group, indicating that lithium ions can be accommodated particularly well within this environment. And this can explain by both the small size of the lithium ions and also by the structure of its hydration shell, where the lithium ions has the very structured first hydration shell that consists of 4 water molecules, so that it has the its hydration shell has the tetrahedral structure. Those taking into account the strong binding of the lithium ions to the phosphate groups of DNA, the one question can be raised, and it's can the local ion DNA interactions influence the DNA organization at the larger scales? And actually, the DNA aggregation is observed in the presence of multivalent ions or the polycations, but whether this effect occur in the presence of the monovalent ions, I would say they still remain the open question. So yeah, inspired by the strong binding affinity of lithium ions to phosphate groups of DNA, we performed atomistic molecular dynamics simulation for the 4 different systems. Each system consists of the 5 fragments of DNA with the same nucleotide sequence shown here. And these 5 molecules, they were immersed in the water box, and the salt in the concentration 1 mole were added. So we added salts of lithium, sodium, and potassium. And also the 4th system that we modeled, it was also with the lithium chloride salt, but in the lower concentration, 0.1 mole. So here's some details of our simulation, and I want to highlight the force fields that we used. And this was the Albert Parham BC1 for the case of DNA, which is shown the good agreement with the experiment in the case of the simulation with DNA. And for the ions, we use the Young-Chetnam. I hope I pronounced it well, in correct way. We used Young-Chetnam parameters, and model of water we used TIP3P. So we calibrated the system for 200 nanoseconds first, and the production run was 400 nanoseconds. It's the chunk of trajectory that we analyzed. So what we did first is that we visually analyze the trajectories that we obtain. And here I'm showing the snapshots for the case of system with lithium and the concentration 1 mole. And yeah, it can be clearly seen that DNA, they approach each other close and form the clustered arrangement. While in the case of the sodium ions and also the potassium ions, the DNA stayed more separated, as you can see, and only the single contacts can be observed. So to quantify what we observed in the trajectories, we calculated first the coordination numbers for the phosphate group belonging to the different DNA macromolecules. So the x-axis on the graph shows the DNA-DNA pair for which we calculated the coordination number, and the colors of the bar, they correspond to the color scheme of the different DNA in the system, just for clarity. So we can clearly see that the system with the lithium chloride, they exhibit the high coordination numbers, indicated the frequent DNA-DNA contacts in the system, and interestingly that we observe the same effect as for the higher concentration of lithium and for the lower concentration of the lithium chloride. And in the contrast, for the system for sodium chloride and with the potassium chloride, they show the much lower coordination numbers if we compare with the lithium system. And this show us that in this system, the DNA association is much weaker. So to understand why it's happening, we also calculated the number of ions around the oxygen atoms of DNA. And just to remind you that one phosphate group, it has two non-bridging oxygen atoms, it's O1 and O2, and O1 atom is exposed to the solution while O2 atom is directed towards the major group. So what this graph shows? This graph shows the time average ion occupancy on the O1 atom of the phosphate group, shown as a y-axis, and the same time average ion occupancy at atom O2 of the phosphate group, shown as x-axis. So this circle on the graph, it corresponds to one phosphate group. So what we can see from the calculated results, that in the case of the lithium system, the occupancy of some of the phosphate groups are close to 1. And this shows us that, first of all, that both oxygen atoms of one phosphate group are bounded to the lithium ions for two different lithium ions. And also, since here it's a time average number, these lithium ions, they bound to these phosphate groups continuously, which is very interesting result, right? And also the occupancy numbers, values, they are shifted towards higher numbers in the case when we increase the concentration in the system. And also what is I want to say and also important is that we do not see the preferable bonding of lithium ions to the O1 or O2 ions. But in the case of the sodium and potassium ions, they are preferably bound to the one oxygen atom of the phosphate group, and it's a rare event where the both oxygen atoms can be coordinated by lithium or by the ions. So yeah, taking into also, sorry, I would mention this may be better now that our analysis also show that in the case, for example, in the system with lithium chloride in 1 mole concentration, the approximately half of the phosphate groups of all the DNA in the system are simultaneously occupied by ions, lithium ions. Yeah, but in this case, only 10% of the phosphate groups in the system are state free, so they are not interact with the lithium ions, which is important point for the future. So taking into account this strong binding of lithium ions to phosphate groups, they can mediate the DNA contacts in the system that we observed during the trajectory. And therefore, we analyzed ion-mediated phosphate crosslinks over the trajectory. And the two oxygen atoms of different phosphate groups we considered to form the crosslink or the contact when they simultaneously coordinated by the same lithium ion. So we distinguished 3 distinct contacts in the system. It's the contacts through atoms O1, O1 of the different phosphate groups, O2, O2, and the mixed contact through the atoms O1, O2 or O2, O1, which is equivalent. So we calculated the probability distribution of number of simultaneously active pairs in the system. And our results show that lithium, in the case of the lithium chloride system, the distribution is broad, indicating the multiple simultaneous active pairs. And with the increase of the concentration of lithium chloride salt, we see that the number of the pairs are also increases. In contrast, for the sodium and potassium system, the zero contact state is dominated, while only 2 or 1 simultaneous crosslink can appear in this system. We also studied the lifetimes or stability of the observed. Sorry, this should move, I guess. Oh, yes, but it doesn't move. Anyway, it should.
Maybe to push play.
I don't know how to do this. No, for some reason it doesn't work. Anyway, it was the nice video. So we calculated the lifetimes of observed crosslink in the system by just providing the frame-by-frame analysis and calculating the duration of the individual event. And here on the graph, you can see, on the slide, you can see the, let's say, the contact maps where the Y and X axis show the residue number of all the DNA in the system. And so you can see between which residues the contact is occur. Anyway, and also I would like to mention that these blocks on the diagonal, they show the residues of DNA that belongs to the same DNA, so no contact here can be observed. So what we can see is that in the case of the lithium system, some of the contacts lifetime, they can reach even the hundreds of the nanoseconds, while in the sodium and potassium, First of all, we see the less number of contacts, and those that do occur, they exist for the tens of the picoseconds. So the results for molecular dynamics, they suggest that DNA contacts can occur through the ion-mediated phosphate links, and to gain the deeper to the to go deeper to these molecular level interactions, to see how stable the complexes that we observe is. So we performed also the DFT calculation for the series of different complexes. We started with the isolated phosphate group, and step by step we added new elements to the complexes. We also compared the different ions in order to see how they can stabilize the phosphate pairs. And this structure, they were extracted from the molecular dynamic simulation, and then they were optimized. So we calculated the total interaction energy for each of these complexes, and our calculation showed that the highest absolute value of total energy is obtained for the case of the complex with lithium ions, showing that it's the most stable complex. So this result is consistent with those that we observe in molecular dynamics, and also they provide the further explanation while the lithium ions, they more efficient in mediating the contacts between the DNA. So in order to determine whether this crosslink can be detected experimentally, we also calculated the vibrational spectra. We calculated both the infrared and the Raman spectra for all the complexes that I showed before. But then we selected only the complexes that we observed in the molecular dynamics simulation, and we combined the spectra of all these complexes in order to obtain the characteristic model spectra of the DNA in lithium chloride solution. So the contribution of these complexes, it was evaluated depending of their population over the simulation trajectory. So we calculated the spectra. This is a spectra in the case of 1 mole lithium chloride. The black line corresponds to the DNA in solution, so without these complexes, which includes crosslinks, and the red line shows the DNA with crosslinks. So one can see that first of all is the intensity in the case of the second spectra with the crosslink, it increases, but what is the important thing is that in the region where we usually observe the backbone vibration according to experiment from the 900 to 1300 inverse centimeter, we see that in the case of the DNA with crosslinks, some of the new peaks appear, and obviously they should characterize the vibration, the formation of crosslinks. And these results can be useful as the spectral markers for the possible experimental detection of the DNA association. So you can ask why can this works at all, and both our MD and DFT calculation, they provide the molecular mechanism of the DNA association in the lithium chloride system. So the first thing is that I repeated once again that the lithium ions, they have the very strong binding affinity to the DNA phosphate groups, and as a result, the this is often situation when the both oxygen atoms of one phosphate group can be simultaneously bound to the lithium ions, and it can be clearly seen from the just a random snapshot from the molecular dynamics simulation. And this lead us to the high binding of the lithium to the phosphate groups, and is as a result the partial compensation of the negative charges of DNA phosphate occurs, and the electrostatic repulsion between the neighboring DNA, it can be reduced in this way, and thus this DNA can approach each other more closely. And the second important thing that the lithium ions, they preserve their tetrahedral hydration structure, even in the case when they interact with the phosphate groups of the DNA. And you can see that the oxygen atoms of the phosphate groups of the different DNA then can replace the two water molecules from the lithium hydration shell, but still they save this coordination geometry of the lithium hydration shell. So both of these factors, it's a strong binding of the lithium ion to DNA, partial charge neutralization, and also the formation of the ion-mediated stable phosphate-phosphate interactions through the where the hydration of lithium ions involved. This all they contribute to the possible DNA association. So let me stop here because I cannot talk anymore. And here is the just several sentences to conclude what I said. The ion hydration shell, it's an integral part of the structure and dynamics of the DNA macromolecule. The canturion hydration governs ion-specific interactions and dynamics around DNA. And the last point, that unique hydration properties of lithium ions and its strong binding affinity to phosphate groups, they both can promote DNA association through stable intramolecular cross-links. So yeah, thank you for your attention, and yes, thank you for the financial support for the National Research Foundation of Ukraine. And yeah, I open to your questions.
Thank you very much. So now, dear colleagues, we have a time for questions. Please, who has question, please raise hands. Yes, Professor Grubisic from University of Belgrade. We are very glad that you are here. Please ask question. Sonia? She unmute.
Okay, sorry.
Yeah.
So, can you hear me?
Yes, please.
Okay, so thank you, Tetiana, for this very nice talk and lecture. So my question is concerning force fields and accuracy. So how much do current DNA force fields can capture the coupling between DNA conformational changes, hydration, and ion binding? Are there some limitations of the current force fields? So are you satisfied with the force field that you use? You mentioned some PB0 force field, as I heard.
Yes, you're right. Thank you for your question. So in general, as far as I know, the force field that we used for DNA, amber force field, it's pretty good to describe the conformational or the changes in the DNA. So there are many works on this force field that show that it's pretty good one. But there is the question about the ion parameters in general, and yeah, it's a open question that ions can overbind to the phosphate groups of the DNA. And actually, there is some works by Aximentiev group, for example, that show that they provide some molecular dynamic studies for the Young-Chetnam parameters and also for the Bogolyubov parameters. And they show that actually ions have show the overbinding to the phosphate groups, and in their works they also show some like condensation. So they can you please switch the slide? Sorry. So thank you. So they propose some parameters or the fixes to the both the Bogolyubov parameters and the Young-Chetnam parameters, and it's called like a Q-fix. And you can see that they specifically propose these changes for the case of Lennard-Jones parameters. So they simply shifted the position of the minimum in the parameters of the Lennard-Jones. But the problem is in this fix, for my opinion, is that they add these changes only in the case of the interactions of ion with the phosphate groups of DNA, while they do not change anything in the ion-water interaction. And those, in this case, we studied this question actually before I have all these graphs. So we can see that this blue line corresponds to the Young-Chetnam parameters. It's a potential of mean force that we calculated. Yes, so the blue line corresponds to the lithium near the phosphate groups from the Young-Chetnam parameter, and red line corresponds to the same, but with this Q-fix from the Aximentiev. And also we compared these potential wells with the results for the lithium together with oxygen atom of water in both that we have for the Young-Chetnam and also the Q-fix, which is identical should be. So you can see that in the case of the Q-fix changes, in the case of lithium interaction with oxygen atom of phosphates, the well, potential well, become much not deeper. In the case shallow, thank you. It become more shallow compared to the Young-Chetnam parameters, while in the case of the lithium with oxygen atoms, it's still deep. So in this case, the lithium ions, it's pretty hard for them to dehydrate and bound to the phosphate groups. So here is also the question if it's correct approach. Yeah, so in short, I'm not,
Short, Nadine Schultz.
no, I don't show here the Nadine Schultz. No,
The previous.
this is not the point here. So in short, yeah, I'm not sure about the parameters, but I mean, we don't have the ideal parameters. It's still the works going on. And anyway, we observed the different behavior for the case the lithium ion, sodium, and potassium. And if even the lithium ions is overbound to the phosphate groups, the results still show that ions interact differently with the DNA in the modeled system. I hope I answered to your question somehow.
Sonia?
Okay, yes, thank you for this detailed answer. So it seems it's really challenging to choose the proper force field or to develop, to slightly refine the existing ones. So thank you, Tetiana.
Yeah,
excellent work. Thank you very much.
Thank you for your question.
Thank you. And we have another question. Polina Konevska, please.
Yeah, thank you. Thank you very much for really fascinating results and the research you made. And I want to ask if do you think that the result you receive about localization lithium next to phosphate groups and creating these clusters with water and DNA clusters. Do you think this compacting lithium next to DNA and like bringing additional water will affect on DNA internal mobility and how DNA can be recognized with other ligands. Its approachability to bind with other ligands.
Regarding the last question about the ligands, as I understand the question correct, actually this situation with the aggregation with lithium ions is like, for me, there is no point to study the additional binding with ligands, because the lithium ions, they are, as I know, they are toxic. I know that now the lithium ions also used in some the treatment of diseases, but in general, I don't see the point to I don't know, bound them with ligands. If only these ligands can promote the additional effects on the condensation.
Yeah. So it is possible for some technological reasons.
So yeah, I'm very slow going to this. So yeah, if you remember yesterday, Sergei Nikolaevich showed that the results on the lithium-ion batteries. So yeah, there are also some work following these lithium-ion batteries. They used some like synthetic polymers together, just added to additional to this structure, and as far as I remember, they like help to create the better.
They create additional crosslinks, and also the question about influence of lithium ions on the internal dynamics. So, as I understand, we wanted to ask about conformational dynamics, something like motion of nucleosides and so on.
Yeah, I have no idea actually.
Well, but I suppose we can expect great influence in this case, because in the case of DNA aggregates, we will have another system. It will influence essentially.
I don't know, the like molecular dynamics method, it's how to say, it's not always show all the possible conformation, and I think so it's a restricted method in this point. So I don't know, maybe you are not agree with me.
Well, I think we can discuss. Okay, other questions? Other questions? There is no raising hands. Okay,
thank you, very nice talk. Still about force field, are the charges, full charges, so lithium is +1 or is it some?
+1. It's a classic force field. So
Yeah.